Hello my fellow genetic lovers and welcome to our channel.
Today, you will be informed about the great wonders of genetics, or the study of how information is passed from parent to offspring,
Including the differences between meiosis and mitosis and who Gregor Mendel was and what he actually did. How to use fun tools called Punnett squares, patterns of inheritance, as well as genetic disorders, and last but definitely not least, drumroll please.
DNA fingerprinting versus karyotyping.
Now if you are ready for this roller coaster ride through the journey of genetics, here we go!
Have you wondered what meiosis really is? Well, we're going to tell you. Meiosis is the process of making cells, also known as gametes, for sexual reproduction. Meiosis occurs in the gonads, which are the testes and the ovaries. Meiosis cells are haploid cells, meaning that they are cells with half the normal number of chromosomes, compared to cells produced in mitosis. Meiosis cells are some of the most unique cells in our body. These cells are unique from their parent cells because during meiosis, the homologous chromosomes, or chromosomes that pair up during meiosis, exchange genes, or factors, passed on from parent to offspring. This process creates unique, random combinations of DNA.
Scientists also refer to this as variation.
The independent assortment of the cells during metaphase 1 also leads to variation. Independent assortment is when each pair of alleles separate independently during gamete formation. Alleles are different versions of genes.
There are two steps in meiosis, meiosis 1 and meiosis 2. In each step, there are 4 phases: prophase, metaphase, anaphase, and telophase.
Before these steps, the sex cells undergo interphase where they can copy their chromosomes and prepare for division.
During prophase 1, the chromosomes pair up with their corresponding chromosomes to form sister chromatids.
Sister chromatids are identical chromatids. Chromatids are half of a chromosome.
Next, crossing over occurs, where genes on the homologous chromosomes switch.
The new recombinant chromatids, which are what chromatids are called after crossing over occurs, now have unique combinations of alleles. During metaphase 1, the chromatids undergo independent assortment, which is when the sister chromatids randomly line up in the center of the cell. During anaphase 1, the homologous chromosomes are pulled apart to opposite ends of the cell. During telophase 1, in cytokinesis, the nuclear membrane reforms, forming 2 daughter haploid cells with 2 sister chromatids. Now, meiosis 2 occurs. In prophase 2, a spindle forms between the centrosome, and the nuclear membrane breaks down. In metaphase 2, the chromosomes line up along the center of the cell. In anaphase 2, the sister chromatids separate and move towards opposite ends of the cell. In telophase 2, in cytokinesis, the nuclear membrane reforms, making 4 haploid cells, with each chromosome having 1 chromatid and each cell coding for different traits. A trait is a genetically determined characteristic. The end result of meiosis is 4 sex cells from the gonads, either sperm or eggs.
Next stop, meiosis versus mitosis.
First, mitosis. Mitosis, also known as the division of a single cell into 2 genetically identical cells, creates almost all of the cells that make you up.
Mitosis occurs everywhere but the gonads. It allows cells to repair and grow, which is one of the reasons why you grow. Lastly, mitosis produces diploid cells, or cells with all the 46 chromosomes.
These cells are called somatic cells, also known as body cells. The cells produced in mitosis are also identical to the parent cells.
Now on to meiosis. Meiosis occurs only in the gonads and makes cells for sexual reproduction. Instead of producing diploid cells like mitosis does, meiosis produces haploid cells, or cells with half the normal number of chromosomes.
The cells produced in meiosis are different from the parent cell.
Carrying on, we're going to talk about Gregor Mendel and the father of genetics. Gregor Mendel was born in 1822 and died 61 short years later, accomplishing so much. Mendel figured out the basics of genetics by breeding pea plants together, and then he observed the traits present in the offspring.
True breeding is when organisms pass on the same phenotypes through the generations.
The phenotype of an organism is the trait expressed by the genotype, such as brown eyes.
The parent generation is called the P generation. The offspring of the P generation are called the F1 or first filial generation, and the offspring of the F1 generation are called the F2 or second filial generation.
His hypothesis was that parents can pass on certain factors, or genes to offsprings that are responsible for inherited traits. Mendel also created 4 rules of genetics.
Mendel's first rule is
Each gene has alleles. So now let's put this information into a real-life scenario. So say you had a flower with purple petals, but other flowers from the same species have white petals. The gene would be the petal color, in this case purple, and the allele will be either purple or white.
Mendel's second rule is that for each trait, an organism gets 2 alleles, one from each parent, which codes for a genotype.
A genotype is a combination of alleles received from different parents and are classified as either heterozygous or homozygous. The organism is homozygous if the 2 alleles from each parent are the same, and is heterozygous if the 2 alleles from each parent are different. Mendel's third rule is that some alleles are dominant and some are recessive.
Recessive alleles can only be expressed if there are 2 recessive alleles.
For example, if you have many people in history, brown eyes are much more common than blue-eyed people, so brown eyes would be dominant, as shown as capital B, and blue eyes would be recessive, as lowercase b. Last, Mendel's fourth rule is the principle of segregation, which states that 2 alleles from each characteristic separate during the formation of gametes, meaning that each gamete only has 1 allele for each trait.
Patterns of inheritance. Here we come. First, let's discuss what patterns of inheritance are. Patterns of inheritance are the ways in which genes are transmitted. Now, to get specific, I will be touching on a few different types of patterns. First up, we have incomplete dominance. Incomplete dominance is heterozygous and is when individuals are a blend of all the possible alleles. An example of this is when you cross a red flower and a white flower, you will get a pink flower. Codominance is heterozygous and is when individuals show traits of both the possible alleles. An example of this is when you cross a brown cow with a white cow, you will get a brown and white cow. On to polygenic traits. Polygenic traits are traits that are determined by more than one gene, and as the genes get more involved, the bigger the range of phenotypes will be. An example of this is human height, which is determined by more than 5 genes from each parent. Next, we have sex-linked traits, or traits determined by the sex chromosomes. And if the inheritance pattern is autosomal dominant, there only needs to be 1 mutated gene for a person to get the disease. It usually occurs in every generation, and it occurs in both males and females. An example would be Huntington's disease. If the pattern is autosomal recessive, there needs to be 2 mutated genes for a person to get the disease. The affected person usually has parents that are heterozygous carriers, and it doesn't occur in every generation. Some examples of this are cystic fibrosis and sickle cell anemia.
In life, you are a combination of your genes and your environment. Your genes allow for a range in characteristics, but your environment determines where you end up on that range. This is known as nature versus nurture. For example, you might live in a sunny location, so the color of your hair might be lighter.
On to Punnett squares, both monohybrid and dihybrid. Yay! Okay, first, what are Punnett squares and why are they used? Well, to answer this question, Punnett squares are diagrams that are used to predict the genotypes of the possible offsprings of 2 individuals.
Monohybrid Punnett squares are with 1 characteristic, and dihybrid Punnett squares are between 2 characteristics.
First, we will learn how to do a monohybrid Punnett square. First, you draw a square and break it up into 4 sections. Then you find the genotypes of the parents. Then you place 1 of the alleles for each genotype above each box. For a real example, say we were crossing a true breeded green pea pod and a true breeding yellow pea pod.
Now, onto dihybrid Punnett squares. You can either use the rule of multiplication, which to calculate the probability of 2 independent events occurring together, you multiply the probability of them occurring separately. You can also use the rule of addition, where to calculate the probability of either or events occurring, you add the probabilities of them occurring separately. Now, you are going to learn about chromosomal versus genetic disorders. Chromosomal and genetic disorders are both disorders that affect the genetic makeup of a person.
Chromosomal disorders are caused by cells with missing or extra chromosomes.
An example of a chromosomal disorder is Down syndrome, where the 21st chromosome is duplicated too many times.
Gene disorders are caused by a change or mutation in a gene. One example of a genetic disorder is sickle cell anemia.
Chromosomal disorders occur because of non-disjunction, which is where chromosomes fail to separate during meiosis. Genetic disorders occur because of a change or mutation in a gene.
Now, we will be addressing karyotypes versus DNA fingerprinting. Karyotypes are diagrams that show chromosomal disorders, or don't show them. They are a picture of a person's chromosomes, and the homologous chromosomes are paired in order from 1 to 23.
DNA fingerprinting is used to compare DNA samples. On it, everyone's DNA is different, like a fingerprint.
It can be used to compare different DNA samples to, and is even used at crime scenes to identify the DNA of criminals.
Thank you for watching. We hope you learned a lot about genetics. Please tune in next week for our next video.